Intermolecular interactions are actually a topic that is studied in an organic chemistry course, and as such they are prerequisite for biochemistry. But because of their importance, I'm going to review that topic and also emphasize aspects of it that are relevant to study of biochemistry. So we'll devote an entire lecture to study of intermolecular interactions. First, we should look at type of interactions that may or may not be classified as intermolecular interactions. Some authors consider them to be intermolecular interactions because those are interactions between different species, actually in this case different ions. Those are ionic interactions. Some consider them that they are not intermolecular interactions because of their strength. They are exceptionally strong. That's effectively ionic bond. So ionic interaction is an ionic bond. So ionic interaction is interaction between oppositely charged ions. Simplest example would be that of sodium chloride, where we have positive sodium cation and negative chloride anion. So they are held together through electrostatic interactions. So that's very strong chemical bond, actually stronger than covalent. And we define in chemistry intermolecular interactions as bonding that is weaker than covalent. This is stronger than covalent. So using that definition, ionic bond doesn't fit into intermolecular interactions. On the other hand, from biochemical point of view, this is clearly an intermolecular interaction because there are numerous ionic interactions that are known between different biomolecules. So this is very important interaction in biochemistry, and it is responsible not only for interactions between different molecules, but also as intramolecular interaction within the same molecule. So oppositely charged parts of the same molecule attract each other and assume certain conformation as a consequence of ionic interaction. Often in biochemistry, such ionic interaction is called salt bridge. And here we have some examples, an example of histidine and aspartate forming a salt bridge, or in another case, lysine and aspartate. You can also see that this can be viewed as hydrogen bonding, which we are going to cover later. So classification of intermolecular interactions is not always that clear. Now, when we strictly speak about intermolecular interactions, those typical intermolecular interactions, they are often called van der Waals forces or van der Waals interactions. So they are defined as interactions between different molecular entities, between atoms, molecules, ions, radicals, or just groups, portions of a molecule. And those are interactions that are weaker than chemical bond, typically weaker than covalent bond. Type of intermolecular interaction depends on type of functional groups that are present. And they are classified as London forces, dipole-dipole interactions, dipole-induced dipole interactions, and hydrogen bonding. There are numerous other types of intermolecular interactions, but we are not going to review them. In general, whenever there is electron density that is involved, that is transferred to some extent from one entity to another, when there is charge separation, there is possibility for intermolecular interaction. There is also additional type of intermolecular interaction, and that's ion-dipole interaction that exists in solution. And that's actually very important interaction for biochemistry. That's very common interaction in biochemistry. A lot of biochemical molecules, molecule of ATP comes to mind. Pretty much any molecule that contains phosphate group exhibits in aqueous solution ion-dipole interactions. So they are actually solubilized through ion-dipole interactions. All of these intermolecular interactions are electrostatic in nature, and in general they are attractive. In biochemistry they are always attractive. They could be repulsive, and that's if species are forced very close together, and then core electrons begin to repel each other. Then electron clouds, or mainly core electrons, begin to repel each other. When that happens, then we call that a steric effect or steric hindrance. Boiling points are often used as a way to measure strength of intermolecular interactions. Concept of intermolecular interactions and various explanations related to intermolecular interactions are theoretical concepts. Boiling points are something practical, something experimental, something that we can measure. Now, boiling means that we are going from condensed state of matter, from liquid, to a vapor or gas, to non-condensed state of matter. In condensed states of matter, and it's liquids and solids, molecules or particles in general are held together through intermolecular interactions. In non-condensed state of matter, the first approximation we consider that means gas or vapor. We consider that there is no intermolecular attraction between those molecules, so that intermolecular interactions are non-existent. It's not quite correct, but the first approximation, that's good enough. Now, gas is a substance that is in form of gas under ordinary conditions. That means ambient temperature, room temperature, and atmospheric pressure. Vapor is a substance that is liquid or solid and has been converted to a gaseous state of matter. So as liquid is heated, molecules acquire energy. They undergo thermal motion. They move faster and faster, and eventually they acquire enough energy to leave the surface of the liquid. And that means intermolecular interactions are broken and molecules leave liquid phase and go into vapor phase. That means that intermolecular forces have to be overcome to go from liquid to vapor phase. The stronger the forces are, the stronger the intermolecular interaction is, more energy is needed, and that means that boiling point is higher. So boiling point is directly proportional to the strength of intermolecular interactions. And for that reason, we use boiling points as a way to estimate or measure strength of intermolecular interactions. Intermolecular interactions that are present in all molecules or all atoms and molecules or particles, all matter, are called London forces, and they are called London forces because they are named after German physicist Fritz London. They are actually the weakest forces, and they are important if molecule is nonpolar. They are due to temporary and induced dipoles. Sometimes they're also called dispersion forces or London dispersion forces. Proper name is simply London forces. Sometimes they're incorrectly also called van der Waals forces, but that's not correct. The term van der Waals forces is used to describe all of the intermolecular interactions. So all intermolecular interactions in general are called van der Waals forces. Intermolecular interactions between nonpolar molecules are London forces. They are important particularly if nonpolar molecules are polarizable. Now, polarizability is ability of a molecule to have its electron cloud distorted. That means that electron cloud is, if molecule is highly polarizable, electron cloud is easily distorted. Due to thermal motion, nuclei and electrons constantly move, and even in non-polar molecule, the temporary dipoles form. And that's simply because the distribution of electrical charge is not symmetrical. If a molecule were at absolute zero, perfectly still, then there would be perfectly symmetrical distribution of electrical charge in non-polar molecule, and there would be no charge separation. But because of thermal motion, there is charge separation, short-lived, very short-lived charge separation, and that generates temporary dipole. That temporary dipole in the neighboring molecule induces a dipole, simply because negative end of temporary dipole attracts the nuclei and repels electron cloud, and positive end attracts electron cloud of the neighboring molecule and repels the nuclei. And so this temporary dipole induces dipole in neighboring molecule or induces dipoles in neighboring molecules. And then these two dipoles or more attract each other through electrostatic interactions. Those interactions are very brief and very weak. Nonetheless, there are lots of them and they add up. So as they add up, sometimes they could even be reasonably strong, particularly if molecule is large and highly polarizable. For example, something like iodine. Then London forces can become strong, but usually they are weak. Here we have example of molecules that exhibit London forces, pentane isomers, and we can see 3 pentane isomers and their respective boiling points. So N-pentane, straight chain pentane, exhibits boiling point of 36 degrees Celsius. The reason why this boiling point is the highest of all pentane isomers is that there is greatest surface area. So it can be in contact with largest number of neighboring pentane molecules and can induce dipoles in largest number of neighboring pentane molecules, so largest number of interactions and strongest London interactions. Isopentane has a little bit smaller surface area, so a bit lower boiling point, 28 degrees Celsius, so weaker set of London intermolecular interactions. Neopentane is spherical, almost a perfect sphere. Sphere has the smallest surface area for given volume, in this case for given number of atoms, and that means that London forces are the weakest, because one molecule of neopentane is in contact with fewest number of molecules of these 3 isomers. Next type of interactions are dipole-dipole interactions. And they are rather strong, particularly strong compared to London interactions. Dipole-dipole interactions are due to permanent dipoles in the molecules. Now, only polar molecules have or exhibit permanent dipoles. That means permanent charge separation. Of course, they are electrostatic in nature. So dipole-dipole interactions, here we have a schematic representation of permanent dipoles, are simply interactions where negative end of one dipole is oriented in such a way that it is in close proximity to as many positive ends of other dipoles as possible, and vice versa. Positive end is surrounded by as many negative ends of other dipoles as possible. Obviously, since they are permanent, those interactions are much stronger compared to London interactions. Average dipole-dipole interaction is about 100 times stronger compared to average London interaction. And here is another way to represent it using those arrows, those crossed arrows, if you recall from organic chemistry, to indicate the direction of the dipole. Now, steric effect or steric hindrance has considerable effect on strength of dipole-dipole interactions because it could prevent effective dipole-dipole interaction. So here we have an example of, let's say, dimethyl ether. In case of dimethyl ether, we have reasonably strong dipole-dipole interactions because negative end of oxygen in a molecule of dimethyl ether, and that would be a free electron pair, can approach carbon, positive end of the dipole, relatively closely. But in case of diethyl ether, interactions are weaker than expected. And that's because that additional methyl group in carbon chain, because of the steric hindrance, prevents interaction of oxygen-free electron pair with positive end of the dipole with carbon atom. And interactions become very weak. On the other hand, if those 2 chains of diethyl ether, two carbon chains, 2 ethyl groups, are folded into a ring to form tetrahydrofurane. So basically, if we take end of each chain, so each chain has methyl group, CH3 group, we remove 1 hydrogen from each carbon and form bond between those 2 carbons, we end up with 5-membered ring. Then there is no steric hindrance because those 2 carbons, 2 methyl groups that would cause steric hindrance are now folded back into a ring. And now we end up with much stronger intermolecular interactions, much stronger dipole-dipole interactions. Simply dipole-dipole interactions are not prevented with steric hindrance. So when you look at boiling points, you can see that diethyl ether has about the same boiling point as pentane. Pentane does not exhibit any dipole-dipole interactions. That's an indication that steric hindrance prevents effective dipole-dipole interactions. On the other hand, tetrahydrofuran, where such steric hindrance is not present, exhibits much stronger boiling point compared to cyclopentane, and that's an indication that intermolecular interactions are much stronger, that in this case dipole-dipole interactions are considerable. In biochemistry, various carbonyl compounds play a very important role. Those could be aldehydes, ketones, or carboxylic acids and their derivatives, in particular amides. They exhibit very strong dipole-dipole interactions, and often those interactions are very important and play a role in secondary and tertiary structure. That means three-dimensional arrangement of proteins, secondary and tertiary structure of proteins, and sometimes some other biomolecules, possibly nucleic acids. Another important molecule is aromatic molecules, such as benzene. And they would appear to be non-polar, but actually they are polar. They exhibit what is called electrical quadrupole. So they have pi electron density above and below the ring, and so that's where negative charge, partial negative charge is concentrated, while along the equator of the ring there is partial positive charge. For that reason, they are capable of considerable dipole-dipole interaction, and that also plays a role in stabilization of proteins, and particularly nucleic acids, and we will actually spend quite a bit of time on secondary structure of DNA, which to an extent is consequence of such interactions. And then when polar and non-polar molecules are mixed, dipole-induced dipole interaction occurs. Simply permanent dipole of polar molecule induces dipole in non-polar molecule, and then the two of them are permanent dipole and induced dipole are attracted to each other and are attracted or held together, at least briefly, through intermolecular interactions, and those are dipole-induced dipole interactions. As one would expect, dipole-induced dipole interactions in terms of strength are between London interactions and dipole-dipole interactions. So they are stronger than typical London interaction, but weaker than typical dipole-dipole interaction. So hydrogen bond is a very interesting type of intermolecular interaction. It is actually a special case of dipole-dipole interaction, but because it is exceptionally strong, it is studied separately. A hydrogen bond occurs only if a highly electronegative atom is involved. And those highly electronegative atoms are nitrogen, oxygen, or fluorine. And those atoms must have, of course, free electron pair. And then that free electron pair interacts with hydrogen attached to another one of those three atoms. So hydrogen is attached to nitrogen, oxygen, or fluorine. So hydrogen bond involves three atoms. So hydrogen has to be attached to nitrogen, oxygen, or fluorine. That's shown here as pi. And then there has to be another atom of nitrogen, oxygen, or fluorine that provides free electron pair for hydrogen bond. Hydrogen bond is indicated by a dashed line here. So on the left, it's shown in general terms, and on the right, specific example of hydrogen bond that is formed between molecule of methanol and molecule of trimethylamine. So in this case, you can see that hydrogen is attached to oxygen, and nitrogen provides free electron pair. This is very strong interaction, and because of that we call it a bond. It's not really a bond. It's not a chemical bond, but because of its strength we refer to it as hydrogen bond. Typical hydrogen bond is about 100 times stronger compared to a typical dipole-dipole interaction. In biochemistry, hydrogen bond is exceptionally important. It's involved in dissolving of various biochemical species in aqueous medium, and aqueous medium is pretty much medium for all of the biochemical processes. Also, it is responsible for secondary and tertiary structure of proteins because hydrogen bond stabilizes segments of protein chains or is involved in long-range interactions between parts of protein chains. It's also responsible for secondary structure and tertiary structure of nucleic acids. So very important interaction. In a hydrogen bond, we can identify hydrogen bond donor, and that's a molecule or part of the molecule that donates hydrogen for hydrogen bonding. So in this example, molecule of methanol would be hydrogen bond donor. Hydrogen bond acceptor is molecule or part of the molecule that provides free electron pair for hydrogen bond. In this case, that would be nitrogen of trimethylamine. And here are some more examples of hydrogen bond. So hydrogen bonds are common among alcohols, carboxylic acids. Of course, water is capable of hydrogen bonding. Among amines, primary and secondary amines are capable of hydrogen bonding, while tertiary amines are not. Tertiary amines do not have hydrogen attached to nitrogen. So on their own, they are capable only of dipole-dipole interactions. But tertiary amines can form hydrogen bond with hydrogen bond donors. So tertiary amines are only hydrogen bond acceptors, and they need to interact with hydrogen bond donor to be capable of forming hydrogen bond. The reason why hydrogen bond is so strong compared to dipole-dipole interactions is that hydrogen does not have core electrons. So if hydrogen is attached to highly electronegative atom, and those would be nitrogen, oxygen, or fluorine, which are three of the most electronegative atoms that there are. There is nothing more electronegative than that. And in fact, chlorine is slightly less electronegative, and it's not electronegative enough, so hydrogen attached to chlorine does not form hydrogen bond, only dipole-dipole interaction. So that shows that atom really has to be highly electronegative for hydrogen bond to occur. So if hydrogen is attached to highly electronegative atom, then electron pair of that bond is strongly attracted by that electronegative atom, and effectively hydrogen is stripped of any electron density and is almost like a bare proton. We only have hydrogen nucleus, almost a bare proton that is exposed to the outside environment. And so that bare proton, since it's not surrounded by core electrons and is stripped of any electron density by more electronegative atom, can approach free electron pair from another molecule very closely and form a very strong interaction, and that's hydrogen bond. Any other atom, like carbon, for example, if it's partially positive end of the dipole, is still surrounded by 1s electrons, core electrons. And so those electrons prevent very close approach to another free electron pair. And so that limits strength of dipole-dipole interaction, and that's why they are much weaker compared to hydrogen bonding. Hydrogen bonds, of course, are additive. So the more hydrogen bonds a molecule can form, the stronger they are. For that reason, hydrogen bonds in water are very strong, and that's why water has unexpectedly high boiling point. Because each molecule of water is capable of forming 4 hydrogen bonds, 2 as hydrogen bond acceptor and 2 as hydrogen bond donor. Primary amines exhibit stronger hydrogen bonding compared to secondary amines, and that's because primary amines can form 3 hydrogen bonds, while a secondary amine can form only 2 hydrogen bonds. Another very important feature of hydrogen bonding is that it could be intramolecular or intermolecular. All of the examples of hydrogen bond we encountered so far were intermolecular hydrogen bonding, examples of intermolecular hydrogen bonding. That means hydrogen bonding between different molecules. But hydrogen bonding between the same molecule, intramolecular, is also possible. Here it's given on an example of simple organic molecule. In biochemistry, intramolecular hydrogen bonding plays exceptionally important role. It's responsible, for example, for stabilization of alpha helix of proteins. It's also responsible for various long-range interactions in protein chains and various long-range interactions in, for example, RNA molecules. So, intramolecular hydrogen bond plays really important role in biochemistry. And finally, hydrogen bond plays very important role in solvation. So when substance or solute is dissolved in a solvent, not just hydrogen bond, but actually any intermolecular interaction. So when a substance is dissolved in a solvent, typically, and we could have any solution, gas in another gas, they're always soluble, but there could be a solution of liquid in gas, or gas in liquid, liquid in solids, gas in solids, and so on. All those things are possible, but typically when we talk about solution, particularly from biochemical point of view, it's solution in a liquid phase. So solids or gas are dissolved in a liquid, or another liquid is mixed with another liquid. Whether substance will be soluble, whether one substance can dissolve in another, depends on strength and nature of their intermolecular interactions. So when substance called solute is dissolved in a solvent, there is interaction between molecules of the solvent and molecules of the solute. So for that to occur, in the process of dissolving, a substance that is being dissolved, called solute, undergoes breaking of intermolecular interactions. So intermolecular interactions between molecules of solute have to be broken. Also, since molecule of solute has to fit in between molecules of solvent in the process of dissolving, that's called solvation, intermolecular interactions between molecules of solvent also have to be broken. So those processes require energy. Breaking of intermolecular interactions between molecules of solute and molecules of solvent require energy. Those intermolecular interactions are replaced by intermolecular interactions between molecules of solute and solvent, and that process releases energy. Now, whether this process is going to be spontaneous or not depends on how much energy is needed to break intermolecular interactions, and how much energy is released in the process. So if intermolecular interactions are about same in strength, so if one set of London interactions is replaced by another set of London interactions, that's a favorable process. And two species will be soluble in each other. That means that non-polar molecules will be soluble in non-polar solvents. If dipole-dipole, one set of dipole-dipole interactions is replaced by another set of dipole-dipole interactions, again, process will be spontaneous, which means that polar species will be soluble in polar solvents. And of course, hydrogen bonding plays a very important role in solvation. So substances that exhibit hydrogen bonding will be readily soluble in solvents that are capable of hydrogen bonding. So species that will be soluble in water, which is the most important solvent for biochemistry, are generally those that are capable of hydrogen bonding or possibly ion-dipole interactions. And here we have an example of intermolecular interactions between molecules of water and alcohol, which means that alcohols in general are soluble in water and water is soluble in alcohol. That means they are miscible in each other. The only caveat here is in case of alcohols, size of this group R. If R group becomes too large, then that gives non-polar character to the entire molecule, and being non-polar, it becomes insoluble in water. So if R is about 8 atoms or more, then alcohol is insoluble in water. Up until about 5 atoms, so if R is up to about 5 carbon atoms, then those alcohols are soluble in water. Between 5 and 8, solubility sharply decreases. By the time we get to 1 octanol, it becomes insoluble. Another factor that affects solubility, as you can see from expression for change in free energy, is entropy. And of course, in case of solvation, entropy is always favorable. So entropy will always favor formation of mixtures. So for that reason, even if thermodynamically, from standpoint of intermolecular interactions, this process is not completely favorable, if it's slightly endothermic, it may still be overall spontaneous process because of the favorable entropy. And finally, interactions that I mentioned earlier that exist only in a solution are ion-dipole interactions. Of course, ion-dipole interactions exist only in solution of polar solvents, and water is polar solvent. Even though primary interaction of molecules of water is hydrogen bonding, water is also capable of dipole-dipole interactions because, of course, it's a dipole. All of the molecules that are capable of hydrogen bonding are also capable of dipole-dipole interactions. And of course, by default, every molecule is capable of London interactions. So, for example, molecule of water or alcohol is capable of hydrogen bonding, dipole-dipole interactions, and London interactions. Ion-dipole interactions occur when ionic compound is dissolved in a solvent such as water in polar solvent. In that case, crystal lattice of ionic compound breaks down, so individual ions leave crystal lattice, and instead of ionic bond, instead of ionic interactions between oppositely charged ions, those interactions are replaced by ion-dipole interactions. So each ion is surrounded by as many oppositely charged ends of the dipole as possible. So here we have an example of dissolving of lithium chloride in water, where we can see that lithium cation is surrounded by 4 molecules of water, where free electron pairs of water interact with a positively charged lithium cation. That would be actually what we call a first solvation shell. Then there are additional solvation shells of water that surround molecule of, or ion of lithium. And we can see that chloride anion is surrounded in first solvation shell by 4 molecules of water, where hydrogens, positive ends of the dipole of water, interact with free electron pairs of chloride anion. Okay, and this completes a presentation on intermolecular interactions.